Hi, everyone, and welcome back to my channel. So in one of my previous videos, I talked about what genomics is. I kind of introduced all the omics actually in one of my previous videos. In this video, I'm going to talk about some specific techniques that are used in the field of genomics. Okay, so I've done my best to find a way to introduce you to these techniques broadly without getting into too much detail. But what I'd like you to understand is what I'm giving you is an overview of these techniques. Underneath each of these techniques, there are lots of different, I guess I would call them specializations, where you take this technique and then you do something different. And these get used depending on what application you are looking to do, right? So which particular aspect of genomics are you looking to research? But I didn't want to go too far down that rabbit hole because, like I said before, genomics is a big field. And if I were to go down into great detail in each of these sort of subspecializations of each of these applications, I think you would get really confused. And so I just want to give you an overview of some of these different techniques that we use when we're doing genomics research. So first, let's start off with epigenomics. So if you recall, epigenomics is what we're looking at in terms of like DNA methylation, histone modifications. We're looking at understanding broadly how epigenetics is affected across the entire genome of a species. And we might do this under normal conditions, or we might do it under certain specific conditions, i.e. being treated with a drug or a cancer model or a disease model, that kind of stuff. The first technique we'll talk about here is something called chromatin immunoprecipitation, followed by PCR. This is abbreviated as qPCR. Like I said, there are lots of different subspecializations of chip. So you might hear of chip on chip or something like that. But I'm going to cover chip with PCR because this is one of the most common techniques used if you're interested in detecting histone modifications. So the way this works is fairly simple. You take a group of cells, you take an organism, for example, it could be an entire fruit fly, it could be an entire nematode, or you take a cluster of cells from a tumor or from cell culture. And what you're going to do is you are going to take that cell in a given state or cells in a given state, and then you're going to use techniques to isolate the DNA protein complexes. All right, so you're going to break those cells open, release all the nucleic acids in there. And then what you're going to do is you're going to treat with formaldehyde. And what this is going to do is it's going to chemically modify so that the protein and and wherever the DNA is connected to the proteins, that those complexes are stuck together. They're essentially chemically glued together. And then what you can do is you can use techniques to shear the genomic DNA. So what that's going to do is it's going to break apart any DNA that's not protected by a protein. So what you're left now is with a collection of DNA protein complexes. All right. Then you can use antibodies that are specific to whichever transcription factor you're interested in, right? So I'm interested in studying what this transcription factor does in cancer cells and how does it differ from normal cells. That's an example of one of these applications, all right? Then what ends up happening is you're going to purify these proteins, and along with that particular protein, you're going to find any DNA transcripts that are still remaining in those DNA protein complexes. You can then release the DNA fragments from those proteins. And then what you're going to do is you're going to perform PCR and do sequencing, PCR to amplify those DNA sequences. And then you're going to take those DNA sequences and map them against the genome. What this is going to do for you is it's going to help you figure out in certain conditions where this transcription factor or transcription factors are binding in the genome. What genes are these particular transcription factors interacting with? And then a lot of times the way we do this is in a differential capacity. We might be saying like, okay, which genes are different in cancer cells as opposed to in normal cells? Or if this particular model mimics a particular disease, is there a difference as to which genes are being interacted with by these transcription factors, depending on which condition, healthy versus a diseased cell? Some advantages to doing qPCR. The first is it's relatively fast. This is something we can get done in a fairly short time scale, which means you can get fairly rapid results, which is sometimes hard to come by in genomics. It's also very well established, which means the protocols for doing this are pretty well established. You don't have to reinvent the wheel here. This is pretty much a gold standard for doing this. So you're not going to get any questions about like, oh, why are you using this technique? How did you apply it? It's already been established in the literature. The other thing that's nice about this is it relies on or allows for genome-wide analysis. You don't need to know which genes you're interested in ahead of time. The disadvantages, you need to have a fairly specific antibody. So if you're interested in a particular protein, in this example, they're using P53, right? So if you're interested in identifying which genes P53 interacts with under a given condition, you need to have a very good P53 antibody, because if it lacks specificity, you're going to pull down other transcription factors. And then you're going to get you're going to be under the impression that certain genes are regulated by P53 even when they're not. Or if you have a poor quality antibody, you might not get all of your P53 and you might miss some of your hits. You might miss some of the genes. The other thing is this: it only detects enrichment abundance, which means you really have to be comparing condition 1 to condition 2, right? You need to say like this is up in cancer cells compared to normal cells. This is down in cancer cells compared to normal cells, that kind of stuff. A related technique is called qRT-PCR, and this allows for single base resolution, which means you can get, and it's more comprehensive, more of the genome gets analyzed. You can detect the difference, for example, between like different transcripts, because you can get very fine resolution in terms of the genes you're looking at. But because it's more specific, your antibodies need to be more specific. It's also much more costly because you're getting more data. It's higher quality data. Quite often what will happen here is you will use this technique to do an initial screen, and then you can follow up with more precise techniques once you have some hits identified, some genes that you're interested in. Now, if you're curious about DNA methylation status, so what we just talked about there was great for detecting protein modifications. What if you're interested in DNA sequencing? Okay, which parts of the DNA are methylated? The way we typically do this is called PCR-based bisulfite sequencing, and this is another great example. You can see here all of these different techniques: clone sequencing, pyrosequencing, mass spec, bead array, all these different things. These all fall under what you do after you do the bisulfite treatment. So the way this particular technique works is it relies on the fact that 5-methylcytosine is the most common form of DNA methylation. I talk about this in my epigenetics conversation, that when we find DNA methylated, it's almost always part of these CpG islands, with the cytosine being methylated on the 5' carbon. All right. Now, if we treat that genomic DNA with bisulfite, any cytosine that isn't methylated, i.e. protected by methylation, deaminates to become a uracil. All right. So it goes from being cytosine to uracil. You can see that reaction right here. Whereas methyl cytosines are protected by that methyl group and they stay as cytosines. Then you follow that up by doing sequencing, and anywhere you detect a cytosine in that sequencing result tells you that that cytosine remained a cytosine, which means it's methylated under whatever condition you're examining on. Now, like I said, there are several more specific applications that can give you better resolution. They can give you more targets as opposed to being, having to know what sequences you're interested in ahead of time. But what you also notice is the cost goes from low to intermediate to high, depending on how much data you want, how good that data is. So again, what money do you have available to you? What questions are you asking? So at the baseline, if we're just talking about like clone sequencing, mass spec, pyro, that kind of stuff, these are cost effective, it's well established, and you can get single base resolution, right? Because you're doing the PCR. So wherever you see a cytosine in that PCR read, you know that must have been methylated prior to the bisulfite treatment, because the rest of the cytosines went away and became uracils. Disadvantages. Well, first off, you need to, because you're going to follow this with sequencing, you need to know the gene or genes, so a whole targeted region, that has to be known. Like, so you notice here, under the baseline, you're looking at about 20 CpGs. Yeah, you kind of need to know what region you're looking at. Now, if you're looking for gene discovery, you're going to have to use one of these more costly approaches following the treatment with bisulfite. The other thing is this: the bisulfite treatment is kind of harsh. And what that means is the DNA can degrade fairly rapidly after being treated with bisulfite. So what we often do here, if we're using this technique, we're going to do this like 10 different times. So if we're looking at a particular genomic region that we're interested in and saying, is there methylation happening under certain conditions, we're going to do this procedure 10, 20 times and get the results to confirm that we get the same results every time, to sort of flush out any background noise or anything we're missing due to this DNA degradation. So you do have to repeat it a bunch of times. But once you've done this, you get very reliable data. Now, in terms of related techniques, there are a bunch of new ones. They allow for gene discovery, so you don't need to know which genes you're looking at ahead of time. There are some that have more specificity, so you get higher quality results. And some of them don't even require the use of bisulfite any longer, which means you get rid of this whole, it's kind of harsh and the DNA degrades. The downside is, at present, they are significantly more expensive. And they're less reliable because these techniques aren't as well established as the bisulfite followed by sequencing. All right. So, and again, this is one of those things that if you're watching this video, 4 or 5 years after I recorded it, some of these might just become industry standards at this point. This might be considered normal for the field. These technologies, they improve very quickly as they get used, which means the costs start to come down and the reliability goes up fairly quickly. So just be aware that you might be seeing some of these techniques become more and more common over the years. All right, so the next field of genomics we'll talk about is transcriptomics. In other words, we're looking at transcripts. We're looking at what happens following transcription. All right, what is the abundance of this particular gene being expressed? The first technique we'll talk about is called serial analysis of gene expression, which gets abbreviated as SAGE. The way this technique works is, first we start by isolating whole RNA. So there are various procedures out there, techniques you can use to isolate RNA over DNA and proteins when you're looking at this. And again, as for all of these, quite often, you're going to be comparing a normal condition to a disease condition. It could be cancer, it could be a chronic illness, or it could be not necessarily a disease, it could be a specific condition, right? Different stages of development following the use of a particular drug, whatever it is that you're looking at. You're going to isolate whole RNA from your samples, and then you're going to purify the messenger RNA from that by using either glass or plastic beads that have a poly-DT tag on them. All right, they're chemically modified to have poly-DT. If you recall, mature eukaryotic mRNAs are going to have a poly-A tail at the end. So we use a poly-DT tag because the A's and the T's will hydrogen bond, and then you can pull down just the messenger RNA, whereas the rest of the RNA can be removed from the sample. So now that we have messenger RNA, in other words, we have transcripts, right? You only get messenger RNA if the genes are active. And now we can start looking at the abundance of cDNA, of messenger RNA in response to that. So the next thing we're going to do after we have our messenger RNA is we're going to use an enzyme called reverse transcriptase. So reverse transcriptase is an enzyme found in some viruses that has the ability to turn mRNA back into DNA. All right. And we've hijacked that for the purposes for this purpose. And we're going to actually see this happen over and over again. And when we convert messenger RNA into DNA, we now call that DNA cDNA. All right, complementary DNA. Why? Because reverse transcriptase reads the messenger RNA, transcribes the DNA that is the complement of that, which, by the way, if we're remembering correctly, that messenger RNA is complementary to the gene that was transcribed. So the cDNA is essentially the same DNA sequence of the gene, except because it's from a mature RNA, there are no introns. So you just essentially have the exonic version of the DNA in the gene that led to the mRNA. Hopefully I'm following this. So you turn genomic DNA into messenger RNA. That pre-mRNA gets modified, all the introns go away, poly-A tail added, 5' cap added. Now we're taking that messenger RNA, reverse transcribing it back into DNA. But because that mRNA is missing all of that intronic information, you don't get the introns included within the gene. You just get the DNA copy of the mRNA. And we do this with primers that have a molecule called biotin attached to that. Why? Because then we can isolate that cDNA from the sample using beads that are coated in streptavidin. Streptavidin will bind to biotin, And then what we know is now, once we pull these off, we now have just the cDNA isolated from our sample. So all of this is an attempt to purify whatever genomic sequences whatever genes are being amplified under this particular condition. The cDNA is then shortened enzymatically to about 11 to 15 nucleotides at length. And then we take them and we can catamerize them. And you can see we take all the SAGE tags and then we shorten them together, all right, into a concatemer. And then we do sequencing on this. And then all you have to do at that point is match those cDNA tags back to the genome. And then you can start looking at relative expression levels. So you can see, for example, gene A, under normal conditions, expressed at around a 9. However, under disease conditions, it's down to a 1.5. This would indicate that this particular gene is transcribed in lower quantities in this disease state as compared to normal. Over here, we can look at gene H and we see the exact opposite. Its expression is significantly higher in the disease state than it is in the normal state. And this might give you an indication. Is this a gene that's misregulated because of a disease? Or maybe its misregulation is contributing to disease. You will have to figure that out through other experiments. So the advantages to SAGE, it's a direct quantitative approach. You don't have any requirement to know what genes you're looking at in the first place. You don't have to have foreknowledge of your targets. You also don't need a lot of RNA. You really don't. And the data analysis is pretty simple. You're going to get your sequence reads back, and then you're going to compare the abundance in under normal conditions compared to the abundance in the disease, the treated, whatever alternate condition you're interested in looking at. Disadvantages, this is relatively low throughput. You're not going to be able to do whole genome-wide analysis quickly with this. So if you're looking for a broader approach, you might want to try one of these other techniques. The next technique we'll talk about is real-time quantitative PCR, also known as RT-qPCR or RT-PCR, depending on which lab you're in and how you want to refer to it. Again, here you're going to see we're going to isolate whole RNA. We will then use reverse transcriptase with poly-DT primers to specifically amplify the mRNA. And I don't have it here, but you are going to convert that in. This is converting it into cDNA again. So again, we have this population of cDNAs. Then what you're going to do is, typically this is done in 96-well format. So you're going to look at a handful of genes in each of these runs. And then you're going to use fluorescently labeled DNA binding probes and perform PCR. And what's going to happen is the machine that does this has like a camera attached. And as more and more PCR product gets made, the level of fluorescence goes up, and the camera and the computer is able to measure the amount of fluorescence that you see. And that fluorescence is directly the result of the abundance of that particular transcript in that particular sample, because PCR just amplifies at an exponential rate. So, you start with a population of 1, and then you go to 2, and then 4, and then, so on and so forth as you go along, right? So what that means is the quicker you reach this threshold, the more of that PCR product you have, which means the more starting material you had. So the amount of starting material in your sample is directly related to how fast the fluorescence accumulates in the sample. And then basically what you do is, again, like you saw, you want to do this relative to a certain sample. So you would do this in, wild type and then compare it to a disease condition or a treated condition. And you would see, is it higher in the disease model or is it higher in the normal conditions? And again, you're looking at relative abundance here. All right. Now, advantages: fast, accurate, sensitive, and you get real-time results. You can get results from a sample in 3 hours. That's about how long, no, maybe actually 2 hours. It takes about 2 hours to do a standard qPCR run here. All right. So when you look at this, you're going to get results fairly quickly. Downside, there is some risk of primer bias. So remember that PCR, the efficiency of PCR is not just related to how much is there. It depends on how good your primers are. So if you have poor primers, you might get better results in some samples than others. And we call this primer bias because it can skew your results. So we want to be a little bit careful of that. So typically, if you find a gene that's very interesting to you, you will redo this experiment with a couple different primer sets to make sure that it's replicated. So for example, if you find a gene is like 10 expressed at a 10-fold higher rate in cancer cells than normal cells, you might want to create a few more primer sets, repeat the experiment, and find out, do you get tenfold every time, or was there some issue with those primers? So, and then you're going to see with all these, there's always going to be some disadvantage to these. Nothing is perfect. And then another technique for transcriptomics is called microarray. So microarray is done on chips. It's actually very interesting. The way you do this, again, isolate whole RNA. Again, you generate cDNA through those poly-DT primers, and then you actually take the two samples and you mix them. And then what you're going to do is a process called hybridization. You take little bits of your sample. You've got these 96 well chips, essentially. And what you're going to do is you're going to put a little bit of your mixed population onto each of these little spaces, and each of these represents a different gene. You then, you have your labeled targets. You prepare it. You mix on the slide. You do the hybridization, the wash, and then you scan. And what you've done is you basically take, let's say that this is your control sample. This is your disease sample. Your controls are one color. Your experimental sample is a different color. And then in this case, they've used red and green. And then what they're going to do is show you, well, green is higher than red in this one. Red is higher than green in this one. And you get your relative abundances. And that's going to tell you whether certain genes are upregulated or downregulated compared to your control under given conditions. All right. So you do get quantitative results out of this. Now, in terms of advantages, again, this is another very well-established technique. So your results aren't necessarily going to be questioned as long as you've done the procedure appropriately. Again, this is an established technique. It is high throughput, and it also uses fluorescence. Some of the older school methods that did something similar use radioactivity. And as you know, whenever you're using radioactivity, there are concerns. Again, this isn't something that's going to give you cancer, but there's a whole other set of clearances you have to get at your university. There's a whole other safety protocols you've got to follow and disposal protocols because you're using radioactivity. The other downside is data analysis can be somewhat complicated because you've got to account for various variables, and it's not something that's straightforward. Often you're going to have to use your functional genomics core or somebody else to analyze these results for you. You're not going to be able to look at the raw data. Somebody's going to have to process this for you, unless you're a microarray expert, and then go ahead and process your own results. And I think this is the last one for this section we'll talk about. This is called RNA-seq. So I talked about ChIP-seq earlier. This is going to skip the chromatin immunoprecipitation step. We're going to isolate whole RNA. We're going to use reverse transcriptase to generate cDNA. We'll assemble our sequence library, and then basically they're going to go ahead, whoever does this for you. This isn't typically something that you do on your own. You need the equipment that's in your functional genomics core if you have one. And you get data that very closely resembles that from microarray, where you're looking at relative abundance. You have to compare condition A to condition B, healthy versus diseased. Okay. Advantages to RNA-seq: it is direct, it is quantitative. You do not have to know any of your targets ahead of time because you're not relying on primers. It works for whole genes. It works for transcripts. You can also discover differences in expression of splice variants. So we talked about how a fair share of eukaryotic genes undergo alternative splicing. This will actually allow you to detect that. And what's really interesting is this technique has gone to the next approach. Newer technologies are allowing you to perform RNA-seq on individual cells, which means you can compare a liver cell to a skin cell, which is a really powerful technique. Again, that's one of those where we're still working out the details. So in a few years, this is probably going to become fairly standard. Right now, it's an exciting new technology. Now, disadvantages. Even though you can detect some splice variants, there can be a challenge in distinguishing between those just because of sequence similarity between the two. But again, we really haven't talked about an approach that's going to allow you to detect these in the first place. So while we're still sussing out some of the details, how to distinguish between different splice variants, it's a technique that allows that. And I lied, there's one more technique, and this is what are called reporter gene fusions. This is a little slow and a little bit cumbersome, but if you want to actually look at expression patterns of genes or the localization of the proteins following gene expression, this is the gold standard. All right. This is more cumbersome. You need to know what gene you're interested in to create these fusions. But you're going to get awesome data on the backside of it. So First, let's talk about the difference between transcriptional and translational fusions. Transcriptional fusions, you're going to take the gene promoter of interest. So I'm interested in this gene. You have to know which gene or genes you're interested in. You're going to take the promoter from this gene. Through molecular cloning, you are going to then attach that to a protein that is fluorescent. In this case, we're using GFP, but there's GFP, YFP, RFP, CFP, lots of different proteins you can use. You're going to take that, and then you're going to find a way to express it in your particular organism of interest. And then what's really interesting is wherever and whenever this gene promoter is activated, your GFP gets expressed. So you can see right here in this transcriptional fusion, these cells in green, that's where this particular protein is expressed. Now notice the green takes up the entire cell. The reason why is it doesn't show protein localization. There's no protein here. You're just saying, hey, when is this gene promoter active? When this gene gets activated, it produces this transgene, this transgenic gene that then expresses GFP as a response. So you're saying, okay, in these cells, GFP is expressed. And you can compare and say it's higher in this cell, in these cells as compared to others. Over here, you can see this is an entirely different promoter fusion. This one's actually translational. We'll talk about that in just a second. The next question we're going to ask ourselves is, okay, well, what if we want to know about the protein? Where in the cell is the protein expressed? Not just when, not just its abundance, but where. This requires a translational fusion. So again, instead of just taking the promoter and fusing it to GFP, we have to take the promoter and the entire coding sequence of the gene and attach GFP to it. Now, when that protein gets made, it's basically your protein with GFP attached to it somewhere, which means now you're getting the entire functional protein made, which means not only are you going to see which cells it's expressed in and when it's expressed, you're going to be able to see where it's expressed. And this is incredibly powerful because now you can detect the subcellular localization of that protein. It can tell you, for example, that this protein is expressed in the mitochondria. It's found only in the nucleus. Here you can see this protein is only expressed in the cell walls of this particular organism. Okay, this again is a very, or I guess it could be the cell membrane, but these are clearly plant cells. This is very powerful because now you can look at co-localization and say these two proteins are expressed together. They're found in the same organelle. They're actually physically attached to each other. Lots of neat experiments you can do. So like I said, reporter gene fusions are the gold standard for measuring gene expression and protein localization. If you can create these in pretty much any molecular biology lab, you can detect these with a fluorescent microscope. So they're easily detectable. There are some disadvantages. First off, you're assuming that you know where all of the upstream regulatory elements are. You're assuming you've gotten the entire gene promoter, and if you miss part of it, you might not get the full expression of the gene expression pattern, okay? The other thing is it's always theoretically possible that the fluorescent tag that you're attaching to the protein could interfere with protein function and localization, all right? So whenever possible, you want to confirm with another version of this protein to make sure that you're not interfering with some key function of this protein or that it is localizing in the same place. But this is usually a very, very good and reliable way to say this is where this gene is expressed and this is how much it's expressed compared to a different condition. Or in the case of the transcriptional or translational fusions, where in the cell is this protein expressed? Or does it go to different locations? So for example, again, comparing a normal model to a disease model, normal conditions, this protein goes in the mitochondria, but when you see a disease state, now it doesn't. Maybe that's a symptom or a sign of the disease. Maybe it's a cause of the disease. Lots of very interesting questions you can ask following looking at a reporter gene fusion. All right, proteomics. What if you're interested in protein expression? One of the most reliable techniques here is called ELISA, so enzyme-linked immunosorbent assay. There are both direct and indirect ELISAs. We'll start with the direct technique. The first thing you're going to do is you're going to take the antigen, okay? What is the thing you're interested in? It's usually a protein. Mobilize it on a 96-well plate. You take your samples, so you generate your samples by purifying the proteins or whatever you're interested in. Out of that, you incubate them in the wells with that immobilized antigen, and then you use an antibody against the antigen to form complexes. And then what you can do is you can look at whether or not these antibody antigen complexes have formed, and that will tell you whether that particular thing is present in your sample, whatever it is that you think is going to interact with your antigen. It could be a protein, it could be a carbohydrate, it could be whatever, but you're using these antibodies to pull down and detect these antibody antigen complexes, and that should give you, first off, is that thing present and relative abundance. In an indirect ELISA, we're going to do this a little bit different. We're going to use a primary antibody first and then a secondary conjugated antibody to detect the first antibody. So here with the direct ELISA, this antibody is conjugated to something like horseradish peroxidase or a fluorescence indicator that will show up when you look for its presence. Here we start with the antibody and the attaching to the mobilized antigen, and then we use a secondary antibody to bind to the first antibody, and that's what we actually detect. That's why it's called an indirect ELISA. You need a secondary antibody, okay? There's also something called a sandwich ELISA, and here you actually begin with the immobilized antibody, okay? Then you put the antigen into the well. The sample goes into the well. Then you use a secondary antibody to detect the antigen, which should only be present if that antigen at first interacted with the initial antibody. So it's just the order in which you do these. Direct ELISAs, we're looking specifically for a conjugated antibody interaction with the immobilized antigen. Second, we're looking at a primary antibody, then we use a secondary antibody to detect the primary antibody. And in the sandwich technique, we actually immobilize the antibody first, then see if the antigen will bind to that antibody, i.e., is it present in the sample? And then we use a secondary antibody to detect that particular antigen. But this is the antigen that is conjugated so we can detect its presence. So the term is basically the way we detect direct, indirect, and sandwich. What gets immobilized and how are you detecting the presence of that antigen? Some advantages, it is highly sensitive and it is highly specific. Why? Because we're looking at antibodies. Antibodies are by definition specific. The disadvantage, sometimes it can be hard to make good antibodies. And if your antibody sucks, it has low specificity, doesn't have good binding strength, well, then you're going to have problems analyzing your ELISA. All right. The next technique we'll talk about is mass spectrometry. So this is interesting. You have to treat your samples, isolate them in a specific way. These are then injected into the spectrometer, and then heat gets used to vaporize those. Electron beams ionize the samples, and then the particles get accelerated through a magnetic field. A readout from a mass spec is going to look something like this. And then we look for abundance. So how high is the peak? And the MZ ratio, how far down the detector before these things come off. All right. And again, to do this, this is one of those things where you need to have preloaded samples. You need to know what this pattern is going to look like. So this is what the pattern looks like for this particular molecule. I actually don't know what molecule that is. So if you're looking for this molecule in your sample, you need to look for this particular pattern when you go through the mass spec, and then you can compare relative abundances by comparing this condition A to condition B. The downside or the advantages, high throughput, and it's very, very accurate in terms of identifying and quantifying the proteins. Downside, you need to have very high quality samples. You need to get rid of stuff that's not meant to be there, or you're going to get artifacts. And you also need to make sure that your samples are homogeneous, evenly mixed, because you're going to have this big sample. You're only going to put a little bit into the machine. And if there's some settling that's going on, or it's not uniformly distributed, you might get bad reads because, your sample isn't mixed in evenly and you pulled off part of the sample that doesn't have an abundance of your particular molecule that you're looking for. All right. And the final way of doing proteomics that we'll talk about today is an old school method. This is called the yeast to hybrid approach. In this, you're going to need two different strains of yeast. You're going to need one that carries your reporter gene and one that builds what we call your prey library. All right. So here's an example of how this gets used. First, what you have to do is you create your strain that has your bait in there. This typically has part of a transcription factor that's required for the expression of a necessary gene. Okay. Oops, sorry. Then you're going to create another library of yeast that has these different preys on there, these different proteins that you think might be attached and attach them to the other half of your transcription factor. Then what's going to happen is if these two are able to interact, if the prey is able to interact with your bait, all right, these two portions. So again, I want to explain this in a little bit more detail. In the first strain, in your bait strain, okay, you have part of the gal4 binding domain, and then attached to that is the bait. It's what we're trying to see will interact with the prey. You then create a library of yeasts that have different prey proteins attached to the other half of the gal4. Okay? Now, if this prey protein can interact with this bait protein, both halves of the gal4 transcription factor will come together, and this will allow the reporter gene to get expressed. This will allow the yeast to grow. If the prey and the bait don't interact, these two halves of the binding domain of the gal4 protein don't come together. Therefore, the gene can't get expressed. So basically what happens is this. If the bait and the prey, the things you think go together, do go together, you will get these gal4 binding domains come together. This necessary gene will get expressed and the yeast will survive. And you can see bait 1 is interacting with these preys 1, 2, 3, 4, and 5, or 1, 2, and 3, not 4 and 5. In each of these cases, this interaction happened. But over here in 4, you can see that there's no yeast growth. That required gene does not get expressed. Okay. And as a result, the yeast doesn't grow. That would be this scenario here. So 1, 2, 3, and 5, we do get the bait and the prey interacting. We do get the rescue, and we get survival of the yeast. In 4, prey number 4 does not interact with the bait protein. The gene does not get expressed, and the yeast dies. Down here with bait 2, none of these interact, and they all die. So you can see the result there. So I hope you're understanding how this works. But again, one of the things you have to understand is you need to know which proteins you're interested in before you do this. This isn't something that you do across the entire proteome of a sample. You need to say, I think these two proteins interact, and here's how I'm going to prove it. So advantages. It's relatively simple. It's not hard to create these things. This is something that's been around for decades. So creating these libraries is not really a big challenge. And it's a great first step for identifying protein-protein interactions. You can do this at relatively high throughput fashion and get a preliminary pass of like, yeah, I think these proteins interact. The downside is you get a relatively high false positive rate. Now, why isn't that a problem? It's not a huge deal because you're going to go in and you're going to perform other studies to show this. For example, you could go make translational fusions of both these proteins and show that they co-localize in a cell when they're expressed. All right. So you do need to do subsequent confirmation. But in terms of a good first pass to look for protein-protein interactions, the Y2H approach is very solid and is very well established. All right. So that's what I have for you for today's video. I hope this gives you a pretty good idea of some very common techniques and how they get utilized to investigate different aspects, whether it's looking at epigenomics, transcriptomics, or proteomics. These are some very commonly used techniques that you're going to find in the field. I don't think anybody would expect you to understand these at the sort of the molecular level at this point, but I do think you should understand how these techniques get used. What questions can you answer using these particular approaches? I hope you enjoyed the video. Feel free to shoot me any comments that you have about it or any of my videos. And I look forward to seeing you again on my page. Talk to you soon.